I'm refuting Denise's claim that the majority of animal tests conducted are unreliable in regards to improved human health. Her secondary claims that I'm going to be refuting are, first, animals are poor test subjects because their physiology, anatomy, and metabolism are different from humans. Second, that 94% of drugs that pass animal tests fail in human clinical trials. And third, that alternatives to animal testing provide more accurate responses in regards to humans. For her first claim, my opponent gives examples of penicillin and morphine being harmful to humans, but have tested well when animal tests were conducted. Now, although morphine and penicillin may result in different reactions from humans as it does in different animals, these are just two specific examples and is not enough information to show that our anatomy is that much different from theirs. Humans react differently to drugs as well, and some side effects may be prominent in some people and different side effects more prominent in others. Even though penicillin and morphine have been proven to not cause the exact same reaction on humans as it does in other animals, the speaker neglected to mention that animal research has contributed to treating conditions such as breast cancer, brain injury, childhood leukemia, cystic fibrosis, malaria, multiple sclerosis, and tuberculosis, as well as pacemakers, cardiac valve substitutes, and anesthetics. Krista B., the director of the University of Texas MD Anderson Cancer Center, has also stated that we would not have vaccines for hepatitis B if it weren't for testing on chimpanzees. Now, as many of you that took biology know, we evolved from chimpanzees and we share 99% of their DNA, making us more susceptible to many of the same conditions and diseases that they're susceptible to. Her secondary claim is that 94% of the drugs that pass animal tests fail in human clinical trials. This data is very misleading when taken out of context. From information I found from the Stem Cell Biology and Developmental Genetics at the Medical Research Council, drugs are tested on animals as well as humans in various different stages. In stage 1, 64% of the drugs tested on animals passed and then went on to human trials. This means that animal tests removed 36% of potential drugs from harming humans that are being tested on. At the same time, 86% of drugs that pass stage 1 in human testing also fail later on in stages of human testing. Yet it was not suggested by the speaker that humans are an inadequate model to test drug development. My opponent also stated that thalidomide was safe when tested on animals, but when used by expecting mothers, it led to birth defects in over 10,000 children. However, she did not mention that the time period that this took place was in the 1950s. Using data that is over 60 years old has many flaws because the way animal tests were conducted back then is much different than they are now. From research that I found from the U.S. Food and Drug Administration, the animal tests done on thalidomide were not accurate because they were never tested on pregnant animals. After being withdrawn from the market, thalidomide was then tested on pregnant animals and, not surprisingly, it resulted in birth defects for their young as well. Fortunately, our medical research has advanced over the past 60 years, and as one would expect, making inaccurate studies such as this one much less likely to occur. My opponent also stated that Vioxx, an arthritis painkiller, was tested to be safe on animals, but ended up resulting in 55,000 fatal heart attacks when released to Americans. The FDA estimated that 80 million prescriptions of this drug were given to the American public, meaning that 0.0007% of Vioxx prescribers ended up dying of a heart attack. The number of animals used in clinical trials is limited for ethical reasons, making it impossible to detect the seriousness of the side effects when such a small fraction was being tested on animals. Vioxx was not a drug that was said to be A-OK for animals and harmful to humans, but more so the drug had such incredibly rare side effects that only massive clinical studies could result in a hope of detecting any of the consequences. My opponent stated as her third claim that animal testing provides more accurate, alternatives to animal testing provide more accurate responses in regards to humans. However, according to the California Biomedical Research Association, in order to evaluate a drug for side effects, a circulatory system is necessary to carry out the medicine to different parts of the organs. Aside from certain isolated experience, such as LD50 tests mentioned by the speaker, the majority of the time, alternatives to animal testing provide no benefits when it comes to testing for conditions such as blindness and high blood pressure. Computer models can only be a reliable form of research when information used from animal testing is done in the first place, which is why I conclude that animal tests are a reliable form of improving human health. Thank you.
All right, proposition and secondary issues are labeled fine. On the first point, the signpost is okay. I think you did a good job explaining the penicillin morphine illustration. I thought you challenged to whether or not there would be sufficiency. And I like the idea, look, there's variation within the species as well as between species. So the idea that one thing is going to discover everything is obviously an unreasonable standard that's being applied there. The example of the hepatitis B test was also pretty effective in showing that it can have a benefit to test on animals prior to testing on human. I thought that the most useful part of your analysis was on the second point, where you're challenging the inference that being made that because a large percentage of drugs tested on humans get rejected after they've been tested on animals was explained pretty effectively. Look, there are thousands of things that are being tested all the time. A lot of them get sorted out, but they get sorted out at different stages, and you explain what those stages are pretty effectively, and I thought that that worked. The interesting example of thalidomide with the follow-up test that said that they found the same thing when they tested on pregnant animals after they had discovered this problem suggests that there's probably a difference in the way the testing methodology works nowadays. I think that that's a reasonable assumption to make. I do think that you could probably demonstrate that without too much difficulty. In the United States, for instance, they also adopted efficacy standards as well as safety standards after the thalidomide experience, and that would be maybe something that you could point to as a way of demonstrating that the animal tests can be integrated effectively into this process. The Vioxx thing, I think there was an interesting statistical point, and there was a good quote that said, look, you couldn't get the results without having a large clinical population that you're applying it to, and that the idea of testing it on animals would have been improbable because it would have required such a huge number of animals to test in the first place. I thought you had a very good explanation of what the reasoning issues on those particular points were. The signpost on the third point needed to be a little bit clearer. First thing, I know that we're talking about circulation, and I think that there is an argument that says that there's an analogous, or there's a need for analogous systems in order to be able to make these inferences. The challenge that says the computer models can only work if we've had prior animal tests, that sounds reasonable to me. I don't know that that's true, so I think there has to be some documentation or further explanation on that point. All right, thank you.